Okay, so if I'm ready, I will start. Okay, so good morning, afternoon, evening. Hola, hello, bonjour. Hola, ni hao, konnichiwa. 안녕하세요. Hello, all. Welcome and thank you for joining us, the 38th seminar in Synthetic Biology Father's Day, I call it today. I am joining SynBYSS from Korea at midnight after my 11th talk in one of Korean companies. I discussed plastic upcycling and climate crisis today. I have 6 more weeks of my crazy 10-week world seminar tour, and I will join you from Cancun, Mexico next week after giving my talk in one of the ECI conferences. So, okay, so it's my absolutely tremendous honor finally to introduce, but very briefly, our pioneer speaker, Professor Jim Collins at MIT. Of course, he does not need any introduction, as we all know him. He is, to me, the father of synthetic biology. Of course, his seminal paper on toggle switch, along with Dr. Eloy's oscillator paper in Nature, are considered as the starting papers of synthetic biology, although many researchers have contributed to the beginning of synthetic biology almost two decades ago. His papers are the reason why I transformed myself from metabolic engineer to synthetic biologist, although there is no distinction nowadays because synthetic biology is broadly defined now, I believe, because many people kind of claiming they are doing synthetic biology. That is some excitement, and then that's another reason I kind of very excited to continue this seminar series. So with that, Professor Collins, the virtual podium is all yours, and thank you so much for your time and pioneering work. I hope you continue to inspire us to, move forward. This field. Thank you so much.
Well, Taisuk, thank you for the invitation, even more so for the really warm introduction. I'm really delighted that you've launched a series and sustained it through hopefully what's the end of the pandemic. And I'd say I'm delighted to be the opening act today for Allison Lopatkin. So Allison, just briefly, actually was a postdoc in my lab for a few years and did absolutely brilliant work. And just by way of anecdote, I actually got to meet Allison when she reached out to me to find out about one of the antibiotic startups that had spun on my lab that she wanted to consider as a job opportunity. And I opened our discussion on asking, why am I not interviewing you for a postdoc position in my lab? And consider myself incredibly fortunate that she took the invite and came in, and you're going to hear absolutely brilliant work. So we'll in for a treat. Turning to just some remarks on synthetic biology, again, really happy, Taisuk, that you're targeting young folks. I think we need to highlight young folks. I think it's a very exciting time in our field. As you allude, we're a little over 2 decades old. I think we really are now moving from being an emerging field to a maturing field. I like to say I wish I was 30 years younger, in that I think it's a tremendous exciting time to be a young person in synthetic biology and systems biology. New technologies emerging, so many exciting, important problems. If I restarted, I would go into synthetic biology for sure, and I would focus in 2 areas. One area that I'm very active in, which is in biomedical research. I think we've seen synthetic biology really come to the fore as part of the pandemic, whether in the context of mRNA vaccines or various synthetic biology, including CRISPR-based diagnostics. I think we continue to have major important challenges to address, where I think we can revolutionize medicine, both with living diagnostics, living therapeutics, and cell-free based systems, both to address infectious disease as well as more complex conditions. And I think prior to the pandemic, I noted at MIT, our young hotshots coming into the school wanting to do life sciences, including synthetic biology, primarily focused on neuroscience and cancer research, areas of great import. But I think the pandemic has highlighted our need to also point our innovation engines to infectious diseases. And I think we responded well, could have responded faster. But I think, as you'll hear from Allison, we have another ongoing crisis to address even in the midst of the pandemic, and that's around antibiotic resistance. Even in the context of the pandemic, antibiotic issues really were at the fore, in that 1 out of 7 people hospitalized with COVID had a bacterial co-infection, and 50% of those who died had a bacterial co-infection. So we need synthetic biology solutions. You know, the second area that I would turn to, and we already heard Tyson refer to it, and that would be using synthetic biology to address issues in climate change and sustainability, whether that be plastic upcycling, to address microplastics in the ocean, whether it be endowing coral reefs with heat tolerance, whether it's figuring out how to better capture and store carbon and or detect various toxins in the environment. I think it's an exciting time. And the challenge there is, how can we really begin to act locally to address more global challenges? Because scale, like we saw 15 years ago in bioenergy, is going to be a big challenge, but should not be a deterrent. I'll just end on, you know, an encouragement for the young folks really thinking about getting deep in this field. Again, tremendously exciting to do. I think as you explore and begin to make commitments, I strongly encourage you to really commit to becoming an expert. I think we have devalued experts, and it's incredibly important to make the commitment to master content and master the skills to be the expert in whichever area of whatever field you decide to choose. And I want to draw a distinction. I don't mean authority. I hate authority. I come from a family of Irish rebels. You know, authority is established and given by social structure. Expertise is earned. And frankly, doesn't need to be recognized, but you need to make that commitment. And the beauty of synthetic biology is being only a little over 2 decades old, you can still get your arms around our literature and become an expert on what's come before, and make a commitment to every day keep up on the literature to know what's happening, so you can innovate and know how your innovation fits. So again, you know, I want to thank Taisuk. I really think synthetic biology will emerge as one of the defining technologies of this century. And with that, I'll hand it back. And again, really excited to hear about Allison's latest work.
Fantastic as always. Oh my God. So I completely agree with you regarding your vision. And I believe your mind and idea are always fresh and young. So you are not old, you are sufficiently young enough. And also, one joyful thing being a professor is that we interact with young people every day. And then that makes us, you know, getting younger and younger every day, I believe. So, you know, but of course, I mean, your vision and your kind of insight and perspective always inspire me and all the young people. And that's why all the young people is excited. And I believe we can solve the problem together, because all the young people will be the leader in the next decade. And then I believe we have the many problems, many, you know, global problem, you know, climate, you know, medicine, all the, you know, food problem. But I believe I'm very optimistic because of the young people in this audience. So thank you so much. And your talk is always kind of fantastic. Thank you so much.
Oh, thank you.
Yeah, thank you. So, okay, so now the main speaker of today with a longer introduction, Professor Allison Lopatkin, you know, basically right now, an assistant professor of computational biology in the biology department of Barnard College of Columbia University. She will be moving to University of Rochester as an assistant professor. And this is a very exciting time to her. And then now basically more teaching focused college to research focused university. So officially, congrats for your new job. And I am excited to see more, you know, your research work, because your work in Jim's lab and Ming Chang Lu's lab is fantastic. I'm kind of so inspired and fascinated by those work. So I'm wanting to see, you know, those work, I mean, the same type of work, I mean, in the future. And she graduated actually from University of Rochester with the bachelor degree in applied mathematics. That's very interesting, kind of transition from mathematics to biological system. And then she got the PhD in Duke University in biomedical engineering under Ling Chung Yu's lab, and then complete her postdoctoral training, as Jim mentioned, at MIT and Broad Institute. Her research and teaching utilize a highly interactive computational and experimental approach. As you can imagine, she's trained as a mathematician, and then now combining experimental approach. And that's very powerful tool, I believe. Not many people kind of combine two things together. And then to quantitatively describe bacterial population dynamics and their implications in a range of clinically and ecologically relevant contexts. And that's a very important keyword here, clinical and ecological here. That means very broad and very hot and important topic she is basically dealing with right now. Research in her lab focuses on investigating mechanisms underwriting the evolution of antibiotic resistance in bacterial populations using a combination of experimental approach, computational modeling, and also bioinformatic analysis. So Allison, please take it away, and it's all yours now. And thank you so much for joining us. Although you will be very busy because of moving, that is not, it's just exciting, but still, very a lot of things to do, but still you are joining us, and thank you so much. And I'm excited to listen to your talk today.
Great. Well, first of all, thank you, Professor Moon, for that introduction and for this entire platform. It's really amazing what you're doing, and I'm extremely thrilled to be part of it. And also to Jim for that also very warm introduction and that anecdote, which truly I think about regularly. It's something I've reflected on a lot, especially as I go from a more mentor teaching focused position into running a research lab. It's incredible how really one conversation can legitimately change the trajectory of your entire life and your entire career. So I have that and more gratitude to thank towards Jim and of course all my other mentors. But as you just heard, I'll be moving my lab to University of Rochester next year, which is my alma mater undergraduate degree. So that's exciting for very similar reasons, that I'll be able to give back to the community that really jump-started my academic career. I started my lab here at Barnard and Columbia in January 2020. So our lab is young, and with all those unfortunate timing, we are still very much focused on fundamentals. But hopefully by the end of this talk, and you'll see also some little tidbits of things I had done in Jim's lab, but by the end of this talk, hopefully you'll see the SynBYO vision of where we're going and what we're trying to do and hoping to do in Rochester. So pretty much all of our work is driven by our fascination with plasmids. That might not seem immediately exciting to a lot of people, since plasmids are one of the oldest and most established tools of synthetic biology. But especially coming from this context of antibiotic resistance, I would argue that biologically, plasmids are surprisingly misunderstood. And practically, they present many limitations that we often accept or overlook that clearly don't pose the same barriers in nature. And so just to name a few that might resonate with some people here, bacteria are not under antibiotic exposure all the time. And yet most of us are still relying on antibiotic selection for plasmid maintenance, which isn't industrially scalable or preferable for living systems. Natural bacteria, even in the absence of antibiotics, can carry upwards of 5 or more plasmids. And yet we still have relatively minimal control over copy numbers or multi-plasmid compatibilities, or ways to introduce orthogonalities with that. Plasmids have shown incredibly long-term stability in the environment. And for us, they continue to cause fitness burdens and escape mutants and things along those lines. And then I would say most importantly, or at least to me, Around 50% of all plasmids are assumed or expected to be transferable through horizontal gene transfer. And only a fraction of that machinery has really been adopted in the research community from a SynBio perspective. And so we really believe that plasmids represent this massively untapped and understudied area for fundamental synthetic biology advances, as well as clinical and environmental applications that are hopefully represented in this sort of overview of the areas we're particularly interested in. But today I really wanted to focus on one of these, which was horizontal gene transfer, and sort of the associated underlying biological intuition or understanding that we're starting to generate. So for anyone unfamiliar, horizontal gene transfer, or HGT, is a type of natural bacterial evolution. And it refers to the lateral transfer of genetic material between potentially unrelated cells. So this is equivalent, the analogy I like to use is it's like I pet my dog and I grow a tail in 3 to 5 minutes, right? Extremely wide species barriers, and yet bacteria do this all the time, regularly. Now, as a scientist and a mathematician, right, this is an extremely natural phenomenon, extremely interesting natural phenomenon. But as an engineer, I really see HGT as a powerful way to leverage natural evolution as a tool for robust genetic communication. And so some of the advantages that I see include things like any sets of genes or gene or sets of genes can be transferred through HGT. And so we have an arsenal of potential functions to use at our fingertips that occurs on quite rapid timescales. So you can think of it as more of a control strategy. It provides systems a certain degree of redundancy by spreading the same functions to multiple members, along with plasticity, since these mobile elements are decoupled from the host genome. And that suggests robustness in response to environmental perturbations. And then most compellingly to me is, as an engineering tool, its ubiquity in nature really suggests some level of generality in terms of context and implementation. And just one example of that is something along the lines of engineering non-model organisms. And so the overarching motivation of what I wanted to use this talk for was really what drives this arm of work in the lab, and that is, can we first understand and then leverage horizontal gene transfer machinery as a synthetic biology tool to effectively engineer our own microbial systems? And so, like I said, we're still focused on fundamentals. So today what I thought I would do is tell you sort of how we've begun to take steps towards this goal and where we're heading and the ongoing projects of what I will be transitioning as I move to Rochester, and all that will lie ahead. And so in this first part, as an applied mathematician, is really to establish that baseline mathematical framework and corresponding rigorous experimental approach to quantify HGT and simulate the dynamics to really try and establish that baseline. Some of this might look familiar to anyone who's seen Meekup talks before. I promise that by the second and third part will be all new information and a lot of unpublished work as well. But I thought it was important to get everyone on the same page. So when I started out thinking about HGT, I found it helpful to have some realistic context to build that baseline understanding. And so to approach this, we looked for natural examples where HGT had been particularly useful to try and better understand the underlying design principles which made it so effective. It turns out that there's really no better case study than the rise and spread of antibiotic resistance. Antibiotic resistance genes have been shown to be widely present on mobile elements. And this map is just giving you a sense of the scale at which a single resistance gene, in this case it's called NDM1, on a mobile element can spread across the globe in a very short time scale. And it did this and continues to do this by jumping through hundreds of different strains and species, starting from a single case, as you can see, over just 10 years, spreading across the globe. And so when we talk about HGT, specifically of antibiotic resistance, we're generally talking about this process called conjugation. And that refers to the transfer of course, plasmids through direct cell-to-cell contact from a donor to a recipient cell, generating what would be called a transconjugate. And so experimentally, we can engineer this pretty readily and simply in the lab. Here we have a transferable plasmid that is expressing GFP and kanamycin resistance. Our recipients are expressing M-cherry and chloramphenicol resistance. And you can see that they're both growing happily and totally fine in this microfluidic chamber until we selected both antibiotics. And what that does is it uniquely kills anything that does not have resistance to both drugs, while selectively now enabling any population or cell that have that unique phenotype or resistance to both drugs to grow. And so that's what you're seeing right now. Those are the transconjugates which grow and overtake the chamber, and they look yellow because it's the GFP M-cherry spectral overlap. And so that video is really capturing what we want a model to do or to simulate. And so when we started this, we really started with what is the simplest possible scenario consisting, or that could describe conjugation. And in this case, the simplest possible scenario is a single population split into two that either does or does not have the same plasmid. So an isogenic strain, right? Either it's carrying a plasmid or it's not. That gives us our state variables. We have plasmid free and plasmid carrying. And they're fundamentally, this can be, or we can describe how they change over time with three kinetic processes. The plasmid free cells can acquire the plasmid through the rate or through conjugation, and that would be described with the rate of plasmid transfer. So I'll be calling that the conjugation efficiency. The plasmid carrying cell can lose a plasmid at some loss rate. That's just the rate that every time a cell divides, there's some probability that it can't, that it doesn't acquire that, or that it loses the plasmid. And then there's the overall growth dynamics of the populations that are present. And in this case, because we're talking about an isogenic population that split into two, either has or does not have the plasmid, we can say they grow at a rate relative to one another, and that would be something known as the fitness cost. So it turns out the two more important parameters here are the conjugation efficiency, or the transfer rate, and the growth dynamics, or the fitness cost. And so we can quantify both of these in the lab very directly and rigorously. And these are just two examples to give you a sense of the magnitude and sort of what we do with them or what it would look like. On the left, we can measure the efficiency or the rate of plasmid transfer. And in doing that, we can start to understand, for example, the types of variables that impact plasmid transfer rates. And so here, for example, I'm showing you that different metabolic effectors can modulate the rate of transfer. So here's conjugation efficiency or transfer rate. Increasing glucose increases the efficiency. Here is just cells harvested at different physiological states also changes the efficiency. On the right, the typical way of capturing plasmid growth dynamics, like I said, is called a fitness cost. And you can see that this presents oftentimes in a reduction in the growth rate. So the red curve is slightly lower than the black one, where those curves are cells growing over time. We can fit those curves to get a growth rate, and taking a ratio of that growth rate captures that fitness cost, which I'm just showing you here. And because this is a relative metric, so it's the ratio, we can tune that fitness cost by making that plasmid more or less burdensome to the cell. And so you can do that by making it, for example, advantageous to carry the plasmid by adding an antibiotic, or by making it more burdensome. The model allows us to generate fairly accurate predictions. And so one of them here was that a costly plasmid can be maintained through conjugation, even without an antibiotic, so almost like a parasite. And at the time, we tested this using an engineered conjugation system, which had a lot of advantages, but the main one was that we had two versions of that exact system, and the main difference was a point mutation, which let the plasmid transfer itself or not. So the fitness costs were the same, everything else was the same. And so we tested that. On the left, we verified that a costly plasmid that was not able to conjugate would be eliminated. And so here you can just see the model is showing you the orange line decays over time, where the y-axis is the fraction of the two population, or the fraction of the population. The plasmid free cells overtake it, and that's because this is a costly plasmid. Experimentally, we confirm that prediction. And then on the right, when you, again, only difference here is exact same plasmid, but now it can conjugate. you can see that the results are drastically different. So that orange line takes over and reaches almost 100%. And so this is showing you that the rate of conjugation here is sufficiently fast to maintain that plasmid despite its costs, right? So on the bottom, there's no antibiotics, but that plasmid is still being maintained pretty robustly over time, simply because conjugation is fast enough. So hopefully you can even sort of hear those SynBio applications immediately, which is you don't necessarily even need to use antibiotic selection to maintain this plasmid at high levels. That's something we have a project focusing on right now, which is fun and exciting, but that's not what I wanted to focus on today. Instead, I just wanted to highlight that this model can be used to scale up the complexity. So I said that we had the simplest possible case. We also can make that more complex. And in this project, we made this as complex as 3 plasmids and 3 species. And again, on the left, the model is predicting the fraction of the population for each species, so yellow, red, and blue. And then the black lines are the plasmid fraction. And on the right-hand side, the experimental predictions were fairly close. So at this point, and that was sort of background, or really just getting us to where the lab is right now, I felt like we had a solid mathematical foundation and understanding of conjugation dynamics. I did a pretty good job. I was able to predict these long-term plasmid outcomes. And so the next step was really improving and refining the quantitative accuracy, because in this sort of SynBio vision, what I really wanted was precise control, which means we really had to understand and have a good basic understanding of the primary drivers of what we were seeing. And so in starting to think about that quantitative accuracy, what I noticed, and this was again around January 2020, was that our model did a really good job of predicting the long-term outcomes of plasmids. But the temporal accuracy was actually quite low in some cases. And so yes, it would predict sort of that outcome in our simpler 3 population, 3 plasmid system. But as soon as we start to get more complex, it probably wouldn't do that great a job. And it certainly couldn't give us accurate understanding of what was happening over time. And that's not what I want from our beautiful HGT controllable system, right? We need accuracy and precision. And so this is where a lot of our work has been focused on. And so this really got me thinking. Fitness costs are well established when we think of growth dynamics. That's something that us and tons of other labs have studied. But the model mismatch really got me thinking that in addition to this long-term fitness cost, perhaps there's another parameter that we have at our disposal for tuning conjugation. And perhaps acquiring a plasmid itself introduces a transient, but not necessarily insignificant, perturbation to the cell. And so maybe acquiring that plasmid is also impacting the growth dynamics, giving rise to that temporal mismatch. But because it's so transient in general, it was not super, in fact, widely understudied when this project started. And what we were basically interested in was establishing a quantitative framework, just like those fitness costs and conjugation rates, to get really accurate, what I will be calling acquisition cost estimates, so we could continue to build out our dynamic models and see if this can improve our temporal predictions. So how do we quantify the cost of acquiring a plasmid? Well, it's actually a hybrid of the 2 methods that I already described. First, we perform conjugation under very specific conditions to generate new transconjugates or de novo transconjugates that have specifically not yet begun to grow. And in parallel, we maintain our adapted transconjugates, or those which we define as having acquired a plasmid and grown for at least 24 hours. So identical populations separated by 24 hours of time. And then we dilute both into the dual antibiotic media, sort of like the video I showed you before, to kill off the parents, add the exact same initial density, and compare the resulting growth curves. And so we did that for the representative plasmid RP4. This is a widely used plasmid that have been used in a bunch of different SynBio applications. The left is showing you that we initiated the growth curves at the same cell density, so those are insignificantly different. And again, keeping in mind that adapted and de novo transconjugates are genetically identical, but transcriptionally, of course, would be different. And on the right, you can see the growth curves. And the newly generated transconjugates have a very clear phenotypic difference where they have a significant growth defect compared to the adapted transconjugates. So the blue curves are much slower with a longer lag. which of course we were able to quantify. So that sort of growth curve was the first indication that we were able to quantify this. I was of course very excited because this was sort of the first senior author project from my lab. But when we saw that, we said, okay, great, what do we do next? We have to quantify it. So on the left here, we have the growth rate. So just to give you a sense, here is the plasmid-free cells. Here's the growth rate of the adapted cells. So this is our fitness cost. Here's the growth rate of our de novo cells. So that's part of our acquisition cost. And then if you take those cells and dilute them 24 hours later, they recover to the adapted level. Similar trends for the lag times, basically just to say that in these de novo transconjugates, both growth rates and lag times seem to be affected. And so based on that, for this particular paper, we derived a quantitative metric that accounts for both of those changes, since we didn't know whether every plasmid would have both one or the other. And so just specifically, bacterial growth curves are really, really nice to work with, of course, they're extremely quantitative, but the time it takes to reach a certain density during exponential phase really only depends on three things: the initial cell density, the lag time, and the growth rate. And so for us specifically, given our two genetically identical populations and given that they're starting at the same cell density, we can just take the ratio of the time it takes to reach some static threshold, and that ratio of times the threshold allow us to capture the magnitude of an acquisition cost. And so we use that to measure 16 plasmids and conditions. You can see that some plasmids, so y-axis is acquisition cost, are extremely low or no costly, not costly at all. And they range in a wide range of costs. And this is independent of the fitness cost, so it's an entirely separate process that is measured independently. Perhaps most importantly for this though, we went back to the model and we incorporated the acquisition costs into our simulations and refit or tried to refit the data that we had collected before. And incorporating these acquisition costs, we found significantly improved the model accuracy. And all you really had to do was add in a transient population, right, that doesn't yet, or that creates a time delay in reaching the full growth rate. And so I want to say or highlight this was led by an undergraduate in my lab, which is even more impressive and amazing. And she's going on to do even more amazing things right now. But since we published that work, we received 2 federal grants to study the mechanistic underpinnings of what's happening. And so just to give you a little sense of what we believe is going on, doing some RNA sequencing. By the time you see a separation in those growth curves, so at that time to threshold, the plasmid gene expression up here has already equilibrated. So there's really no difference in the gene expression levels from the plasmid expressed genes. However, to get to that steady state plasmid profile, the strains that have just acquired a plasmid need to work a whole lot harder. So you can see here, this is just PCA plots of the genome, the genomic differential gene expression. So distinct between adapted and de novo. And when you look at which processes are significantly upregulated, you can see that most are involved in anaerobic respiration and fermentation related processes, highlighting inefficient metabolic growth immediately following plasmid acquisition. And so this is really exciting to see because it suggests that you can tune acquisition costs by altering metabolic efficiency of the cell. And I'm sure Jim is even hearing where this is going because all of the work in his lab was looking at metabolic effects of resistance. And this is definitely where I'm headed in a few slides. But on the left, you can see that we tested this idea, which is increasing metabolic efficiency, which you can do using experimental conditions in a way that non-linearly or non-monotonically changes the growth rate. So basically here we're increasing efficiency, but growth rate is not increasing consistently. And on the right, as you increase the metabolic efficiency, you consistently increase the acquisition cost. It looks like it's a decrease because the time to threshold is an increasing number and it's normalized to the first data point. So I apologize for that confusion, but higher efficiency cells have higher acquisition costs. The intuition behind that is as you force cells to grow more efficiently, they're less able to adapt to the new plasmid by utilizing those inefficient pathways. And so that was sort of self-consistent and came full circle, which is really exciting to see. Now in the last 10 minutes or so, to wrap up today, and especially since I'm going to be moving my lab to an engineering department, which, as I've said a few times now and you heard earlier, I'm extremely excited about. I thought that I would highlight some of the ongoing projects that are right now happening, along with a lot of the unpublished work that we have, mostly because I'm also excited about that, but also to give everyone a sense of where we're going and how we think we'll be able to translate a lot of this information into more SynBio-like tools. When we think about horizontal gene transfer, specifically from a SynBio perspective, what we basically want to be able to control or to do is to control each of these main kinetic processes in a modular way, almost like a BioBrick-like toolbox. And so the main goal of what we're trying to do now is to understand and decouple how each of these components or processes on the left, how they're driven by different components and how they're going to function in different contexts. And so these are sort of the processes on the left and the things we have to tune and engineer them with on the right. And I wanted to finish this talk by highlighting a few of these different connections and their corresponding projects, so you can sort of see how we're going about doing this. So the first one I wanted to share is focusing on establishing plasmid-specific genetic determinants that drive these acquisition costs. Now, in general, if you couldn't tell, acquisition costs are something that we're really excited about. We were the first lab to really do a thorough rigorous quantification of it, demonstrating the phenomenon exists. And we see it as a way to rapidly tune and change conjugation dynamics in systems. But you can also imagine from an antimicrobial resistance perspective, if we can make plasmids exceptionally hard, almost impossible to acquire, then we can slow or even interfere with the spread of new resistance. And so since that publication, we went back immediately to the drawing board to improve our quantitative methods and our throughput. And so it was a little tedious having to calibrate that initial cell density rate to make those curves start at the same exact or insignificantly different cell densities so we could compare those time to thresholds. We've now established a higher throughput method of measuring acquisition costs. So same exact protocol up here, except now we plate directly onto dual antibiotic agar. And that allows us to track single colonies that are growing when we place those agar plates onto a flatbed scanner in a temperature-controlled incubator. The main benefit here is that single colonies we can assume originate from a single cell. And so as long as we're tracking those single colonies, then we can use those directly to compare the times to threshold. Currently, we have measured around a panel of 60 plasmids, or almost 70 now, under identical conditions, and now adding on to those other antibiotic conditions, but at least 60 plus plasmids, where the only difference is the plasmid itself, and everything else is exactly the same. So same recipient, same growth conditions, et cetera. And we're using those to identify potential genetic specific determinants, because right now we don't entirely know what is causing the acquisition costs. And so you can see on the left, this is just giving you a sense of what the raw data looks like off the scanner. X-axis is time, y-axis is density, which is really pixel intensity. Blue curves are adapted and red curves are de novo. So almost immediately you can see some plasmids are just completely no cost to acquire. It acquires it and it is effectively no difference in the cell. Whereas other plasmids have very high costs, very clear phenotypes. And just looking at this data itself is always striking to me, because you can sort of separate them into those buckets or those categories. And so this is an ongoing project where we are continuously adding plasmids to our panel. So if you have a plasmid that you're particularly interested in, feel free to contact us, and we're happy to measure it. But some early results that I thought I'd share to give you a sense of where we think this is headed. All of these plasmids we have fully sequenced and completed, and developed an in-house plasmid annotation pipeline in order to functionally annotate the things that are on each of these plasmids and run what we're doing now, partial least squared regressions, to identify categories that are particularly strong at predicting these costs. And so this is just one example I thought I'd highlight here, that we found replication control modules consistently and strongly antagonize acquisition costs. And it turns out that when you mutate some of these to disrupt control, you can see a noticeable shift. So here are two different replication mutants that have basically higher copy numbers. And in both cases, they significantly increase the cost. And I know that maybe 1 to 1.5 doesn't look super significant, but here you can see the clear biological meaning behind that, which is a clear shift in the curves. And so that's where this project is. Again, this is an effort in acquiring measuring as many plasmids as we can, likely to drive machine learning efforts in order to make these predictions more specific. After that, the second area that I thought I would highlight is very much comes from all of the work that I had done at Jim's lab and all of the many work that Jim and everyone in his lab had done long before me. And this relates to what we can do or think about in terms of modulating plasmid fitness. And so without going into too much detail, my work from Jim's lab had identified a suite of mutations in metabolic genes that were really prevalent in clinical pathogens. And so that's here on the left, where the x-axis here is a bunch of different mutations. Some of these were new mutations we had found in metabolic genes, and those are highlighted in yellow. And some of these were classic expected mutations highlighted in blue. And we had looked in the genomes of 7,000 or over 7,000 E. coli pathogens. And that's what the y-axis here is, how many of those pathogens had that particular mutation. And you can see that our metabolic mutations in yellow span a very wide range of prevalencies and rival those at similar and sometimes greater levels of these classic mutations, or the ones we knew already. We also found that overexpressing the genes that these mutations were found in and these mutations themselves conferred resistance. So on the right-hand side, this is just showing you the MIC, or the change in resistance. And these metabolic genes were particularly important in affecting the resistance phenotype. And so given the extent to which antibiotic resistance genes are on plasmids, and given that now we had found and shown that metabolic genes were involved in a separate, even additional class of antibiotic resistance, this has naturally led us to ask the question here in my lab now, which is whether plasmid-encoded metabolic genes are also prevalent, and do they also confer resistance? Right, and if so, then there's a lot of implications there, but even one of them is, do these also serve as selection markers on plasmids? And so we surveyed over 2,000 published plasmid sequences, and we found that more plasmids encoded at least one metabolic gene than an antimicrobial resistance gene. So just overall, and I should say these were 2,000 plasmids that were able to be transferred via conjugation, so either conjugative plasmids or mobile plasmids. But this is just showing you all of the plasmids and sorted by percentage of gene content. And when you sort it by percentage with metabolic genes, there were more plasmids overall with at least one metabolic gene. These were present in diverse metabolic pathways, similar to the mutations that we had found. And as a first pass of trying to select which metabolic genes to characterize, we looked at their associations with known antibiotic resistance genes. And so just looking at these top three, we could see that, for example, YFBR, really, really prevalent on plasmids, but always co-occurred with an antibiotic resistance gene. In contrast, LPXM and CATG, really, really prevalent on plasmids also, but tended to, not just tended to, were significantly disassociated with resistance genes. So every time one of those genes was there, a resistance gene was nowhere to be found. So we thought those ones were good ones to start out with in terms of testing. And we started to test the phenotypic effect of those genes, and we saw that indeed these genes which are disassociated are shifting IC50 curves. So green and purple are CATG and LPXM compared to the control of the other ones. This paper is under revisions right now, again led by an extraordinary undergraduate student, Lana Palomino, who is going to be medical school bound very, very shortly. And as part of revisions, we are testing a whole bunch of other dissociated genes, and I can tell you already, and seeing the data come in, that the trends are being upheld. So it's pretty exciting. The last thing that I wanted to highlight, and this one's really short, but this one I just think is extremely cool, and I think there's probably people out there that will likely agree, is we're looking at how different recipient strains serve as plasmid acceptors. And so especially in the context of microbiome engineering with horizontal gene transfer, the really main thought process here was we'd like to understand how, or have some understanding of where plasmids will spread and what barriers will block them most effectively. And so here what we're doing is we're measuring conjugation efficiencies for panels of clinical isolates. So here's showing you a subset of 40 Klebsiella pneumoniae and E. coli isolates. X-axis is the conjugation rate for one plasmid, and the y-axis is the conjugation rate for a totally unrelated plasmid. And so you can see almost immediately that some strains, or every dot is a different recipient strain. Some strains are what I've been calling universally bad recipients. Right? These strains are just really, really bad at receiving this plasmid and this plasmid. And for 5 other plasmids, they remain really, really poor. Likewise, these strains up here are really good, high efficiency, remain really, really high. And so there is a lot of effort in the lab. We have many, many students now working on increasing the data set size so that we can use machine learning based understanding of the genomic contributors or explanations for this. Because to me, at least what this suggests is some fundamental first principles level explanation, which once we understand what it is, can ideally leverage into some inducible methods of controlling plasmid transfer. And so just to bring it back to the beginning, I've shown you first sort of establishing that baseline understanding, next improving that quantitative accuracy with that acquisition cost data, and then finally sort of where we're expanding and applying our understanding of HGT into bigger data sets. And this is really the long-term vision that I am extremely excited to kick into here at Rochester. But of course, none of that would be possible at all without my current amazing lab of undergraduates and lab technicians, along with our collaborators who have shared plasmids and strains and thoughts and ideas. And there's really no way to thank Jim and Ling Chang enough, but they have been unparalleled mentors to me in my career, and my gratitude to them truly knows no bounds. So I guess finally, I will be recruiting next year for graduate students. So if you or anyone you know might be interested in this work, please tell them to get in touch. And again, thank you so much to Professor Moon for this wonderful opportunity, and I'm happy to take any questions.
Fantastic. I love it. So this is a lot of work with a lot of young undergrad students, that is even more fascinating. So I have question, but I believe Jim should have challenging question or comment on your work. So Jim, do you have anything?
So Allison, really brilliant work. I'm curious, from a synthetic biology standpoint, given that we so often use high copy plasmids to test out our circuits, what do you think we should keep in mind when we do that in light of the really nice work you're showing on the cost these things impose? And I say it in that also the backdrop that it's intriguing. I think back to the first decade of the field, we really thought of orthogonality as only being limited to sequence homology without really appreciating the burden we were putting on these cells from a metabolic standpoint. And your work clearly points to that we really are disrupting the host organisms considerably.
So that's exactly right. I think that, and this is a lot of where my mind is heading, I think that there are obviously very well-established plasmid replicons and copy numbers that everyone goes to for their cloning plasmids and high expression levels and things like that. And that is just such a minor percentage of all of the replicons that exist in natural microbes and communities. And I just think that we've used those and they work fine, but it should be a very strong effort of, now that we have all the metagenomics and all of those samples stored, to really go back, do more functional analysis of what's there to look at. I am sure, and I guess I'm almost sure, and I would be shocked if we couldn't find better replicons that had lower costs, depending on the context and the species or the strain that you're looking at. It seems that they're so prevalent, and it's not random. There's a lot of rhyme and reason to where certain plasmids and strains become associated, depending on the context and the environment, that we can improve a lot of what we're doing. I'm guessing a lot of that will require machine learning and AI-like efforts, like I know you were describing earlier, in order to really optimize everything that we're working with. Did that answer your question?
It does, thanks. Yeah, very insightful.
Yeah, that's wonderful. So I have a little bit silly question because I could not find an answer to this question for many years. So the question is basically each lab is bleaching cells to kill them before dumping into the sink every day, literally. Of course, this is done by all the bio lab, not only SynBYSS, but all the bio lab in the world. And of course, this is a typical practice for biocontainment, but I have been wondering whether the released DNA, especially with the antibiotic resistant markers, would be safe in terms of horizontal gene transfer, because some bacterial native strain in the environment are very good at taking up DNA from the environment by natural transformation, and then that DNA can be transferred to other bacteria by horizontal gene transfer. So I tried to find the paper, but I couldn't. And have you or others ever modeled such a system or such a process, or done any experiment to predict or mimic this kind of crazy kind of situation? And but we kind of take it for granted, but I believe that might be a huge problem. That might be also contribute to the antibiotic resistant spread, I believe. And so I just curious, I mean, you've done something.
Yeah, so this is also a really great question. So the short answer is that there are people in the QMRA world, like the quantitative microbial risk assessment world, that do things like exposure risks and bacterial levels and necessary parameters to reduce the log reduction of bacteria in the environment. In fact, one of my collaborators here, Carrie Hamilton, is doing that. And for her, she is interested in this exact thing that you're describing, which is how do we incorporate horizontal gene transfer into our microbial risk assessments? And so there's sort of preliminary work there. Certainly extracellular DNA is something that has been thought about in that context a little bit, probably not nearly as much as it should. However, what I can say, more specifically to this is, in general, it's thought that conjugation is much more important for the spread of resistance than transformation, only because it's not going extracellular DNA won't be as stable, and the concentration of bacteria in the water is going to be low. So there's sort of a high probability or low probability of all the things aligning to actually have what you're describing occur. Not to say it's not impossible, it certainly could be, and especially as you biofilm accumulation around pipes and things like that, it makes it more likely. But the evidence right now, and this can always be disproved at a later time, and I will be waiting to see that, and then I'll be wrong, but right now is that transformation doesn't play as large a role at all. So I guess that itself might suggest it's not as important, but that's not to say it doesn't happen, and the ones that do happen very well might be. So I think it's largely understudied, and there should be more people working on it.
I see. That makes sense. So thank you so much. So let me check with people's question in the chat and then Q&A. That means your talk is perfect in a sense. So I actually we only have the 3-minute left, so I will conclude in that way. We have more informal chat if somebody wants to join us. So thank all for joining and staying today. We will meet again next week on June 23rd, Thursday, the same time, the same Zoom link. But I will be joining from Cancun, Mexico. I will be more normal time than midnight after my 16th talk in I think Cancun, and then we'll have the Professor Jay Kisling, my academic grandpa and a former NSF SynBYSS director at UC Berkeley and JBay, and Professor Andrew Jones at Miami University. He is basically the young speaker for the next week. And as usual, the follow-up informal chat will occur without recording. So please stay here if you are interested in chatting with today's speakers. I'll promote you to panelists who can speak and show your handsome and pretty face. So thank you so much for joining us, and I stop recording. Let me stop, in that way we freely chat.